The world of laboratory science has improved tremendously owing to the advances in medical research and technology. With this in mind, one can hardly imagine a healthcare system without the contributions of clinical laboratory technologists. Laboratory professionals play a crucial role in the detection, diagnosis, and treatment of the disease. Having said so, it is as important for these clinical laboratory professionals to be qualified, competent, and of course, licensed and/or certified. The latter part mostly becomes the stumbling block for some as they try to challenge the examinations without the necessary tools. And quite honestly, there are no shortcuts or quick solutions to passing your CLS or MLT examinations. So making a study plan and complete preparation is critical to your success. And this is where ACTS Review Center specializes. Our review program is quite unique compared to others. Why? Well, besides the regular services that we offer like review materials, question bank, online learning tools, and our accomplished and excellent team of instructors, we conduct our review in a more personalized level. We make sure that we are always available to assist them in any way we can. Yes, it is indeed our commitment to serve them. And that is why this year, besides the traditional or the classroom review that we offer, we are introducing our online review course, which would benefit a lot of candidates who cannot avail of our traditional courses. Now, this is no ordinary online review because we combine the expertise of our instructors and the power of technology. So all throughout the online program, you will have video stream lectures with multimedia presentations and medical animations. Watch this. The two important groups of phagocytes are the macrophages and the neutrophils, in which, as mentioned earlier, participate in host defense by engulfing and destroying microorganisms or antigens. The sequence of events in phagocytosis includes the following. First is recognition of the antigen. Second is attachment of the antigen, followed by ingestion, and then finally, destruction of the antigen with molecules that are cytotoxic to the antigen.
In the next video, note that phagocytes can interact directly with microorganisms via receptors that recognize molecules present in the surface of microorganisms. Phagocytes have also been shown to interact indirectly with microorganisms through opsonization. Opsonization is the process of coating the bacterium to enhance phagocytosis. The microorganism is then engulfed by the phagocyte into a vesicle known as phagosome. The phagosome is then taken inside the macrophage where it fuses to another vesicle termed as lysosome. The lysosome contains powerful chemicals and enzymes which can destroy bacteria. Now, the fusion of the phagosome with the lysosome forms the phagolysosome. Inside the phagolysosome, the microorganism is then killed and digested. Finally, the digested materials are eliminated from the macrophage by the process of exocytosis.
Our next topic is the evaluation of liver function. The liver is the largest and most complex internal organ. It is the major site for the synthesis of plasma proteins, endogenous lipids and lipoproteins, especially cholesterol, coagulation factors, glycogen, and glucose. It is a critical component of the reticuloendothelial system involved in the storage of energy, and a very important endocrine organ. The liver can be viewed both macroscopically and microscopically. The microscopic view of the liver reveals a bilobe organ richly vascularized with two main supply vessels, the hepatic artery and the portal vein. The microscopic, structural, and functional unit of the liver is the lobule, which consists of, first, the cords or the hepatocytes that surround the central vein. Second, the sinusoids, consisting of blood spaces lined with endothelial cells and Kupffer cells that surround the cords which drain into a central vein. And third, the bile canaliculi, or small channels between hepatocytes that carry bile formed by the hepatocytes to the bile ducts. The hepatic physiology depends on the components of the liver.
Hemostasis. Hemostasis is derived from the Greek word, which means stop of blood flow. The main goals of hemostasis are, number one, to keep blood in its fluid state by keeping a balance between procoagulants and regulatory factors. And number two, to arrest bleeding through the activation of the different coagulation factors. When there is injury to a blood vessel, primary hemostasis takes place.
The first thing that happens is vasoconstriction to reduce blood flow to the injured area. Then the platelets will adhere to the injured surface of the blood vessel. They will release their granule contents like serotonin and adenosine diphosphate. Adenosine diphosphate leads to further aggregation of the platelets. The aggregated platelets will blend into one homogeneous mass to form a platelet plug or hemostatic plug. Secondary hemostasis also takes place. It involves a series of blood-protein reactions that results in the formation of an insoluble fibrin clot.
So there you go. Just keep browsing our site for more information. We wish you success with your examination and with your career as clinical laboratory professionals.